Right, come on then. Yeah, welcome. Welcome.
Thank you.
Right, so we'll carry on. Now, again, take a deep breath, get your oxygen into your brain, and now we're going to have three speakers, three scientific programs. Okay, yours first.
We get to the topic.
Yeah, perfect.
Good day to everyone. It's a great pleasure to be here. And yes, indeed, I'm a Drosophilist, a geneticist actually, working on Drosophila. But from the very end, I would say that I'm also implicated in what is called basic or fundamental science. So I'm trying to get to the root of the problem. I try to understand what is behind the phenomenon, what is the genetic regulation of any kind of phenomenon. So today I'm going to talk about the implication of Cocoon in cellular metabolism. So basically, the cells are going to be the topic of my discussion. How I came across the Cocoon story, I would like to apologize for the old logo, but this is telling to you that indeed we are working on now for a couple of years. So indeed, Dr. Lyons and Dr. Salkai came to me and tried to convince me that here it is a water, would you be so kind to test it to see if it does something to your Drosophilas? And then being a water, you know, I wasn't too enthusiastic about checking a water because a water is a water, I was thinking at that time. So I did it. And just to get the idea of the experiment, it's quite a simple one. Let's try to raise the flies on a media that would contain normal water, shown here in red, and then on a media in which, you know, the normal water is replaced with the Cocoon water, and then just assess the lifespan of the flies. And as you can see, there is a difference between the red and blue line, and the blue line and this difference would indicate that the flies raised on the Cocoon water would live a bit longer. So actually the Cocoon water could rise the average lifespan of the Drosophila. We repeated this experiment a couple of times. The same results came. We came across the same result. We said, okay, this is quite interesting. So from now on, I'm gonna start looking on what exactly the Cocoon is doing. I also have to tell you that my lab is very much implicated in what would be the natural remedies, how the natural remedies would act on a molecular level, on a cellular level. What are the molecular mechanisms behind, you know, natural remedies like extracts made of plants and so on, especially plants. So, nice enough, we were very happy with these kind of results. But certainly being a geneticist, you know, we tend to simplify things around us, and we would see everything, any kind of phenomena, you know, as the sum of the interactions between genes and environmental factors. Again, about environmental factors, one can think about nutrition and Cocoon. And this is what we started to combine, nutrition with Cocoon. And let's check what's going to be the influence of nutrition and Cocoon, and this influence, how it's going to manifest at the level of the life quality of Drosophila. So when it comes about life quality, one can assess it at the level of the whole individual. Yeah, that's it's basically known by everyone that this is kind of homeostasis type of effect. But you can also assess homeostasis at the level of cells, and that's what we are doing. So I'm not very much interested in what is going on at the level of the whole individual, but on what is going on a cellular level. This is because my whole background is about the genetic basis of cancer. So this is a kind of slide that is telling you that assessing homeostasis at the multicellular organism level would require, you know, a kind of studies that would tell something about the responses that are generated through neuronal and hormonal type of responses, and that would generate a kind of steady state. So I'm pretty sure Robert already mentioned a couple of results regarding what is going on this level, and today there will be other more lectures addressing this issue, how the Cocoon interferes with the homeostasis at the level of a whole individual. While regarding the cellular level, we know for sure that the cells can sense, they know somehow what is their nutrient and energetic status. We just don't know how they know these things, but they know. And regarding their nutrient and energetic statuses, they can trigger, responses that are very much related to the metabolic profile of the cell. It's very much related about how the redox potential is regulated, and also how the mitochondrial homeostasis or the mitochondrial activity is regulated in the cell. And then guess, all these kind of responses should be matched with the cell cycle control. We don't know how these things are achieved, but the cell is working wonderfully. And the cell, to our knowledge, is the best working, as the most efficiently working open system in this universe. So getting back to the study, to the topic of our studies, certainly we knew that there was a wonderful biochemist, Otto Warburg, you already mentioned, Robert, who in the 1920s and 1930s started to study the cell's metabolism. And basically, he could differentiate, he could define 3 types of metabolic profiles. One is a kind of metabolic profile that occurs in the case of cells that have plenty of oxygen, we would say in normoxic conditions. And basically, in such situations, the glucose is going to be transferred into pyruvate. The pyruvate is going to enter the mitochondria. So glycolysis is coupled to what is the oxidative phosphorylation. The cells would generate a lot of ATP, and the cell would be really happy to live on wonderfully. However, when differentiated cells run out of oxygen, there is no oxygen, we would say in anoxic conditions, basically this glycolysis is going to get uncoupled from what is going on in the mitochondria, in the oxidative phosphorylation, and a lot less ATP is going to get produced, and the cell tries to survive up until the energetic background would allow it. So this is a kind of metabolic profile termed anaerobic glycolysis. However, when Warburg was assessing the proliferative tissues in quite normoxic conditions or the tumor cells in hypoxic conditions, he realized that a part of the pyruvate obtained through the glycolysis is going to get turned away, the majority of the pyruvate is going to get turned away from the mitochondrial type of oxidative phosphorylation. A wee bit is going to enter, is going to get burnt actually. So a bit more ATP is going to get produced, and the cell is going to live on and try to survive. Later on, certainly, more studies have been carried on these kind of metabolic profiles. The last one, called aerobic glycolysis, or as a sign of recognition termed the Warburg effect, was shown to produce enough ATP in order to let the cells survive. Also, this type of metabolism would allow the cells to protect themselves against reactive oxygen species or oxidative stress, and also would allow the cells to produce enough macromolecules in order to support, the proliferation of these kind of cells. So this is basically the strategy for the cancerous cells to survive. Later on, the geneticists came into this story, and they tried to assess how this kind of metabolic profile is set. And basically, they were able to define a kind of genes like the PKM2 or the HIF1 or the MYC or the P53 genes being implicated in setting up this kind of metabolism. Certainly, it turned out that all these genes are also implicated in the cancer. When these genes are going to get mutated, then the cancer is getting instated in the cells. So getting a bit more into details, and I really apologize for you for these kind of details, but you will understand what we are talking about. So here it is, what is going on in the so-called Warburg effect type of metabolism. The glycose entered the cells, and in the cytoplasm, the glycolysis is initiated. Basically, it's going to reach the stage of pyruvate, and then all of a sudden, the pyruvate is not going to enter the mitochondria, but it's going to get transformed into lactate. A bit earlier, I was showing you this protein, the PKM2, which is encoded by the gene that it normally is functioning in the muscle cells, but due to some mutation, this gene is going to get ectopically activated in cancerous cells. So we have a situation when a gene is working exactly where it shouldn't work. And that's how basically the whole system is going to get, the whole glycolysis is going to get modified. Actually, the turnover of this process is going to get slowed down. And because it's going to get slowed down, then the system is going to allow other processes to get attached to this system. So actually the glucose 6-phosphate is going to be used to produce nucleotides. These kind of intermediates is going to be used to produce nucleotides and amino acids. And this one to produce glycerol. So actually, this is the way how basically the cancerous cell's metabolism cheats on the cell. Actually, by modifying the metabolic profile, would produce nucleotides, amino acids, glycerol, so actually proteins, the building blocks of proteins and lipids, in order to get the cell surviving, in order to prepare the cancerous cell for the next division. So it's not going to use these the glycose to produce energy, but instead of producing energy, it's going to use it to make it building blocks. So it's a kind of trick. Then the trick, it goes even further. Why? Because the lactate that is produced is going to leave the cells, enter the blood, and then through the blood reaches the liver, and through the liver, in the liver, actually the lactate is going to be used to make glucose, and the glucose is again through the blood is going to get back to the cell. So you see actually the system through this so-called Cori cycle is going to become self-sustainable. Up until, you know, there is glucose in the system, it can go on, it can fuel actually the cancerous cells themselves. Certainly, this is the kind of biochemistry behind the whole events. But then scientists were curious, okay, we know now what are the players, but what are, if there are any external signals that would trigger these kind of events, these kind of modifications regarding the metabolic profile? And it turned out that hypoxia is the so-called environmental effect that would make a cancerous cell to become even more strict on switching its metabolism. And the first thing that you can see in the case of hypoxia is that basically the ATP utilization in the cells is going to get reduced. Why? Because a lot of reactive oxygen species are going to get produced when hypoxia is first instated, and this reactive oxygen species would shut down translation, the last step in protein synthesis that would require a lot of energy, and on the other hand would shut down the functioning of the sodium-potassium type of ATPases that again would use a lot of ATP. So that's the first thing that would happen in hypoxia. That's the first reaction. The cells would reduce its ATP utilization. But later on, a bit later on, you see the hypoxia is going to induce the activation of HIF1. HIF1 is a transcriptional factor that once activated is going to enter in the nucleus and is going to activate the expression of several hundreds of genes. And among these genes, there are some that would encode for instance, microRNA 220, which is a kind of molecule that would reduce the activity of the mitochondrial electron transfer chain. And by doing that, certainly the ATP production is going to get scaled down. HIF1 alone can, not alone, through the action of, by inducing other enzymes like the inducible nitric oxide synthase, again would reduce the complex 4 activity, or it can induce other changes at the level of cyclooxygenases that again would modify, you know, the whole respiratory chain functioning. And all these kind of events eventually is going to lead to a really reduced ATP production. However, this is not the only effect of the HIF1 activated situation, because there are other genes whose expression is going to increase substantially, like the lactate dehydrogenase, that would make the pyruvate to turn away from the mitochondria and get transformed into lactate, Or on the other level, so it's a kind of double control as you can see in here, the HIF1 would induce the expression of another gene, PDK1, that in turn is going to inhibit the pyruvate dehydrogenase activity. That is meant in normal normoxia type of conditions to facilitate the pyruvate entering the mitochondria. So as you can see, the cell, through the action of the HIF1, would generate not just one effect, but several effects. And that's, again, a very important message. When we talk about the Cocoon water, we should bear in our minds that several mechanisms would be triggered, and all these kind of mechanisms should be looked at together in order to get a more proper picture of what the Cocoon is doing. Certainly, from all these kind of stories, you could imagine that basically the HIF1 is a very important regulator of the hypoxia, is basically the trigger that would make the cell to switch to what is a kind of strategy to survive hypoxia. And no wonder, that scientists across the world were looking for molecules, for remedies, to somehow to block the activity of the HIF1. Why? Because blocking the HIF1, for instance, in the case of hypoxia in a cancer situation, would make the cells not to survive, but to commit to enter apoptosis and to die. So it would be the cure, a kind of cure for cancer. Certainly, in normoxic conditions, you see the HIF1, again, it can be controlled by other proteins like the PhDs. This is not about doctoral degree, but it looks nice. And most of the studies across the world also highlighted the implication of HIF1 and hypoxia in many other cellular events. I was talking up until now only about how HIF1 is implicated in regulating the metabolic profile, but you can see there are many other issues like the immune response. Robert also mentioned the immune response a bit earlier, but it's also genomic stability and cancer and so on and so on. Just to give you another hint about how things are interrelated in the cell, here you can see basically the HIF1 getting activated. But HIF1, once it's going to get activated, it can activate another pathway, which is the NF-kappa beta pathway, which is implicated in inflammation. And once this one is activated, it can further fuel the HIF1 activity. So this slide is telling us that basically HIF1 is going hand by hand with inflammation. And indeed, there have been a lot of studies shown that hypoxic conditions can generate inflammation, while basically inflammation can generate hypoxic conditions. So these two things, hypoxia and inflammation, are somehow interrelated. And this is the case also in cancer. So to find a cure for cancer, you should address the issue of inflammation too. Or why not, I can just tell you in advance that it's also a good idea if you want to fight obesity, you should address the issue of inflammation. As you can see in here, it's a kind of table that shows all the genes that get activated through the action of HIF1. You see some of them are implicated in the cell's growth and survival, glucose metabolism and so on, so many others in what is cancer about. So one single protein inside the cell can activate several genes, and these genes, through their action, would mediate the survival in hypoxic conditions and also in inflammatory conditions. Now we are turning to the flies. One would say that why flies? Flies because we are studying flies because flies are the best known multicellular organisms, and this is where we can combine all our knowledge and we can study the genetic basis of any kind of life-related phenomena. And for a geneticist, it's pretty obvious, and I would try to convince you to accept the idea that everything that goes on in our body, even at this moment, is controlled by our genes through the expression of the genes and the mechanisms that are generated through the cooperation of genes. In Drosophila, we know for sure that every single gene would interact with many other gene-encoded proteins. So we can create a kind of interaction map for every single gene-encoded protein. So here you see a kind of simplified version of an interactome, where basically is HIF1 is shown. But when we did this kind of interactome study, we learned that basically next to the HIF1, there is another gene, the PhD, the propyl hydroxylase, that I was showing you a bit earlier, that regulates, that makes inactive the HIF1 in normoxic conditions. That is also a very important player. Why? Because it would interact with many other proteins known to being implicated in cellular growth and division and survival and so on. So we said we should try to address this issue by looking not only at HIF1, but also all the other players, and try to assess what would be the effect of Cocoon on all these genes. Why? Because we imagine this kind of metabolic switch that occurs from normoxic conditions to hypoxic conditions as a double switch, in which HIF1 is just one player, while the propyl hydroxylase is another player. And we know from other studies that the propyl hydroxylase is a kind of enzyme that could monitor the oxygen level inside the cells. So this is the protein that senses the oxygen level. So if there is hypoxia, then this protein is not going to get any more active. So if it's not active, then HIF1 is going to turn into maximum activity. But this is an amazing protein. Why? Because it can also monitor the C vitamin level inside the cell. So you were telling a bit earlier, Robert, that the C vitamin is important. Yeah, it's important, and it's great that your studies are showing that it's not just the only oxygen, but the C vitamin is also important. But the protein that would sense this inside the cell, the oxygen and the C vitamin, is the propyl hydroxylase. And we are addressing its implication too in this whole process. So we were checking out what's going to happen to the flies if we are going to shut down the cyclin, the propyl hydroxylase, and other players like cyclin D, E2C, and the von Hippel-Lindau type of genes. These shown in red are also very well-known oncogenes. In most of the human cancerous cells, these genes are also mutated. Turning it down them, in the eyes of the Drosophila would result in a really affected phenotype. You see the whole structure of the eye is going to get affected, and because we have the same phenotype, that's a good indication that all these genes are indeed collaborating with each other, cooperating with each other in order to fulfill their functions. But this is not, and to give you one more hint, when we check the level of the phenotype, the manifestation level or the penetrance of the phenotype, this mutant phenotype, in the Cocoon water, these kind of effects were greatly reduced, saying that the Cocoon water can counteract the effects of the mutations. We should get more into the details to see what are the compensatory mechanisms in here, but that's for sure some other time. It could be an interesting topic. Next, what we have done with Drosophila is to look at the development of the fly. Why? Because the development of the fly is really strictly timed. So we know that the embryonic stage would last for a day, and then it comes the larval stage, and that would last for basically 6 days, and then it comes the pupal stage, basically 4 or 5 days, and then the new adults would emerge, and everything would start all over again. And we know that this kind of timing depends on certain environmental cues. Initially, when the embryogenesis goes on, basically the embryo is sealed off totally from the environment. It's just the exchange of gases occurs. So, yeah, certainly oxygen is there. But no nutrient uptake is going to happen. So the embryo is going to live on what the mother put into the egg when the egg matured. And when we were looking at the metabolic profiles, we found interesting different kind of metabolic profiles across the Drosophila development. So you see, in the early stage, we have a situation, a kind of normoxic type of situation when glycolysis is coupled with the oxidative phosphorylation. This is the moment when basically the carbs are burnt in order to produce energy. The same thing would happen for the fat. Lipolysis and beta oxidation are at height, so the fat is burnt in order to get some energy. Because there is no environmental intake, there is no nutrient intake in here, we are presuming that there is some kind of genetic program that would govern this kind of metabolic profile in here. Why? Because a couple of hours later, so started from mid to late embryogenesis, you see glycolysis is going to get uncoupled from the oxidative phosphorylation. This is a kind of Warburg effect. So, basically, the carbohydrates are not going to get burnt anymore, but they are going to be used to synthesize nucleotides, amino acids, and the glycerol in order to promote the survival of the cells. I would like to pinpoint that in here, basically, the oxygen level does not increase or does not decrease. So I would say that it's a kind of normoxic situation. So you can have this kind of uncoupling of glycolysis from the oxidative phosphorylation even in normoxic conditions. So then again, we would think that there is a kind of genetic control in place that would regulate these issues. However, when the larvae would hatch, they would start eating a lot. So there will be a nutrient intake, and this nutrient intake, would change again the metabolic profile. Why? The aerobic glycolysis is going on, but lipolysis and beta oxidation is going to go down, and then the fat is not going to get burnt anymore, but stored. And you see, this is how the larvae manage to grow. So they would use the carbs and the fats that they would eat, in order to build up their bodies, to grow, to become bigger. Why? Why? Because once they reach the so-called critical mass checkpoint, can you see it? That's a very interesting checkpoint recently identified in the case of animals. It's multicellular animals. It would tell the animal when to switch its whole neuronal and hormonal status in order to get fully matured for sexual reproduction. We know where exactly is this checkpoint acting in Drosophila, and again, it's something really amazing that we can relate the functioning of this checkpoint to the nutrient availability and cell cycle control. This is one of the few cases that we know for sure that the cell cycle is controlled through the nutrient intake. So you see it's quite a complex picture, but we have all these kind of metabolic profiles all together. All we have to do is just to feed the Drosophila and then assess what is the timing of the life cycle. So we did this, and I'm getting to the end of the story. But the most interesting part comes now. So look at the situation when we were feeding the flies, with on a normal sugar, normal water media. You would see that the length of the embryonal and larval stage would last for 7 days. When we replace the normal water with Cocoon, so normal sugar media as the normal media, and we replace the normal water with Cocoon, we would have the same type of developmental timing, saying that the Cocoon water is not doing any harm, going back to the cytotoxic effect or not. So there is no cytotoxic effect, and I can tell you that there is no genotoxic effect too. Why? Because we have the same length for the embryonic and larval development. However, when we did another experiment, so we used a kind of high sugar media with normal water, then the length or the duration of the embryonal and larval period increased substantially from 7 to 10 days. So why is this, what is this happening in here? I'm going to get back to this a bit later. However, when we replace this normal water from the high sugar media with the Cocoon water, it's quite amazing that the length of the embryonal and larval development decreased substantially. Basically, it's 8 days. So 1 day longer compared to the normal situation, or 2 days shorter compared to the high sugar normal water situation, saying that the Cocoon water can somehow reduce the negative effects of the high sugar type of media. What is going on in a high sugar? This was shown in the case of Drosophila, that if we put Drosophila on a high sugar diet, that would induce a type 2 diabetes where hyperglycemia, insulin resistance develops, and certainly the levels of the 3-glycerides and 3 fatty acids is going to increase. And there is a lot of inflammation going on, and pure larvae struggle there to cope with all these kind of problems. And this is why, their developmental time increases. There have been done 2 microarray studies in order to see what kind of gene expression is induced up on a high sugar diet, and it turned out that the glucose transport, metabolism, lipid synthesis, and storage, oxidative stress involved genes are expressed, not normally but abnormally. So that would be the molecular mechanisms behind, the high sugar-induced type 2 diabetes. And as you can see with our Cocoon, with this wonderful Cocoon, we could reduce these kind of effects. We are very much into these type of experiments. Get back to you soon. We were also curious about what the Cocoon is going to do in a situation when we would induce hypoxia. And when we induce hypoxia, in a normal sugar, normal water conditions, then the survival rate would be not more than 22%. However, when we replace the normal water with the Cocoon water, the survival rate would increase to 84%. So that's a substantial difference. When larvae kept on high sugar media and normal water are exposed to a hypoxic shock, then basically the survival rate is even lower. However, when we replace the normal water in the high sugar media with the Cocoon water, the survival rate is going to increase again. So what is this experiment telling us? It's telling us that basically the Cocoon, it's not only that it can contracurate to some extent the high sugar induced problems, but also can cover, what is even more important, can cover for the hypoxia-induced lethality. This is a kind of picture showing you what is really happening during the experiments. Here you can see, some kind of larvae that were subjected to a hypoxic shock on a normal media with normal water. So actually, when they are subjected to a hypoxic shock, they would crawl out, from the media. While in here you can see the Cocoon water containing media. In here they would remain in the media and some of them would go out, but then later on in a couple of minutes they would go back and they would continue feeding. Why? Because the Cocoon water is a kind of source of oxygen that would help them to cover for the missing oxygen from this environment. Actually, at this moment, we are very much implicated in finding out what is going on in these 2 situations. So what is the genetic control behind this kind of shift that occurs, in order to reduce the side effects of high sugar media? And this is a kind of table that shows you what we are assessing, and we are assessing the expression level of the whole genes in the Drosophila. This is a kind of formerly called microarray type of experiments. These days we would say it's a total messenger RNA sequencing. We are waiting for the results. It's a tremendous amount of data, but certainly it would let us know what are the mechanisms behind these kind of compensatory effects of the Cocoon water. And we are very excited. So to get to the end of the whole story, we are applying a kind of system biology type of approach where we would combine different kind of experimental systems based on Drosophila, like high sugar, normal water, Cocoon water type of issues. And then we are assessing on the level of transcriptomics what is going on. And then bioinformatics is the next step, where we would sort out all the information generated through transcriptomics in order to be able to define what is the implication of Cocoon in conditions like diabetes inflammation. With this being said, I would like to thank for your attention, and I'm ready to take questions if you wish.
Excellent.
Like.
Excellent.
Thank you.
Here you are. So we have brought science into this meeting. Now what we're going to do, hydrate ourselves, get oxygen into and put some water and come back. So we're going to have a break for, say, 15 minutes. Come to the same place, please.